Welcome to my presentation on membrane structure and function. Name is Ricardo Williams, biology teacher. So today I'm going to go through the structure and function of the membrane. Specifically, we're going to be looking at the fluid mosaic membrane model of biological membrane structure. And we are also going to explain the processes of diffusion, facilitated diffusion, osmosis, active transport, endocytosis, and exocytosis. So to begin with, every cell is surrounded by a membrane, sometimes called a plasma membrane or cell surface membrane. By now we're more familiar with the term cell membrane, right? So membranes are also found inside the cell where they envelope organelles. Examples of this include the nucleus, chloroplast, and mitochondria. Now, the function of the plasma membrane is basically to separate the internal environment of the cell from the external environment of the cell. So as a result of that, we could say that the cell membrane or the plasma membrane acts as a barrier. It also is a partially permeable membrane, and as a result it regulates what enters and what leaves the cell. So therefore it acts as a regulator. Good, so we have two main functions. One acts as a barrier, two act as a regulator. Alright. So the structure of the cell membrane, it has mainly four major components. First one, the phospholipid bilayer. We have peripheral / extrinsic proteins. We have integral proteins, and we could call them intrinsic proteins. And that very important molecule down there, we call it cholesterol. So we have four major structures, and we'll be looking at them individually now. So the phospholipid bilayer, by now you should recall from a previous presentation I did where I looked at lipids, and we looked at examples of lipids. One example looked at was triglyceride, and then we also looked at an example called phospholipid, right? So it is a lipid that has a phosphate head attached to the glycerol head, and that makes it very unique. And the uniqueness we're referring to here is that the phospholipid bilayer has a double layer of phospholipids. Now the phospholipids consist of an electrically charged glycerol head and two fatty acid tails. So in the example I mentioned before, when we looked at lipids and looked at triglycerides, we had three fatty acid tails, but this phospholipid now has two fatty acid tails, and the reason for that is because one of the fatty acid tails is replaced by a phosphate group. That phosphate group is overall has a negative charge on it because of those oxygen atoms there. They are very highly electronegative, and therefore it gives the glycerol head a negative charge. So what we end up having is a phosphate, not a phosphate, but a glycerol head with a charge, and the fatty acid tails that have no charge. So in a sense, the phospholipid is bipolar because it has a charge head and a zero charge fatty acid tail. So the electrically charged heads are being described as hydrophilic, and what we mean by hydrophilic is that it is water loving. When we say it's water loving, we mean that it has a charge, so therefore water can attach to it or form a bond with it, because you know that water is dipolar. It has a positive and a negative charge, weak, but they are there, and so they are always attracted to any molecule that displays a charge on it, whether it be positive or negative, right? The fatty acid tails, however, are going to be described as hydrophobic. The reason for that is because they have no charge, and because they have no charge, water won't be attracted to them. So in a sense, they are repelled. Good. So that's why we say, if you look at the liposome, mycelium, and bilayer sheet, if you notice, the tails are all bundled together. So even in the bilayer here, you see the tails turn to each other in the center, and the heads point in the same direction. Good. So that's how we end up with a bilayer. Good. So when you look and you see a droplet of oil in water, sometimes when you're washing up the dishes, or any form of grease that comes in contact with water, it generally forms that little globule or little circular shape, and this is the reason for it, because the lipids will turn in a specific way, because the tails are zero charge, so they won't react with the water. Alright, so moving on to the proteins now. So but the lipids, they make up majority of the membrane. So as you can see, the example down here, the bilayer sheet, this is basically how the structure of the membrane is. This is like the foundation. So everything else that we will discuss will fall within this structure here. Alright, it will become clearer as we move on. So the proteins basically are in two groups. We have intrinsic and we have extrinsic, right? So the extrinsic proteins are those that will be found on the surface of the bilayer. Good. And the intrinsic proteins are those that will be found within the bilayer, right? You have some that are embedded but not going all the way through, and then you have some that span all the way through the membrane, and these we call them transmembrane proteins. An example of that is channel proteins. So if you look here on this example, starting back from the foundation, here we have in red all the phospholipids. So you have a bilayer, means you have one layer of phospholipids on the top, and then another layer of phospholipids at the bottom. Good. This is what the membrane will look like. Also, look at we have here, we call it integral protein. Is the same as the transmembrane protein or the intrinsic protein. Once the protein is embedded right through the membrane, we call it intrinsic or integral or transmembrane protein. Here we have a peripheral protein, which means that it is just stuck in the surface. That surface can be inside the cytoplasm of the cell or on the extracellular surface of the cell. Whichever position it falls, it will be peripheral, meaning that it is on the surface. So an example of a channel protein is found here. It's a protein that is we call it transmembrane or integral as well, but it acts as a pore, a channel, a hole through which things pass. Good. Additionally, there are some other molecules to be included, and we call these glycoproteins and glycolipids. Now, glycoproteins is just those protein molecules that have a chain of carbohydrates attached to it. Good. And glycolipids is just those lipids that have a chain of carbohydrate attached to it. Good. So that's the only thing that makes it unique, and so we give it the name glycoprotein or glycolipid. I'm looking for a glycolipid. Let's see any yet. No. Well, anywhere you see a phospholipid molecule with a carbohydrate chain attached to it, or a chain of sugars attached to it, then it is a glycolipid. Okay, yes, here we have a glycolipid right here. Good. So this is a carbohydrate chain attached to a lipid molecule. So glycoproteins, basically what these do, these are like some IDs on the cell, and they act as receptor sites, allowing specific molecules from outside of the cell, such as hormones, to bind to them, right? And they act as recognition sites because of their specific shape. They are specific to particular types of cell. They bind proteins in neighboring cell membranes, holding cells together, and this is important basically in actually forming tissues, because remember, cells, specific cells group together to form tissues, right? So these glycoproteins help to hold each cell together to help to form tissues, right? Glycolipids are basically serving almost the same function. They actually, yes, deals with cell recognition and binding cells together to form tissues. Good. So glycolipids and glycoproteins are basically similar, right? Now, cholesterol is important because within the phospholipid bilayer, one of the most important nature about the bilayer is that this thing is always in motion. It is not rigid, it is not fixed, it is not stationary, it is in motion. And when we're saying it is in motion, we don't mean that it is running all over the place. We just mean that this thing has a slight fluidity to it. Good, which means that the molecules themselves kind of move around and shift around a little bit. So cholesterol helps to maintain the fluidity in the bilayer and prevents the phospholipid molecules from coming too close together during low temperatures, because you know when things get cold, right, the particles lose energy, so they vibrate less, so they come closer together, right? So the cholesterol now kind of act as a buffer in between, so when the molecules tend to come closer together at low temperatures, it prevents them from coming too close. And in higher temperatures, when they have a lot of energy and they tend to vibrate and move very far apart, the cholesterol now acts as a buffer again and kind of hold them together, that they don't go too far apart. Because if they go too far apart, then you know that the membrane itself will be broken down, because the entire structure depends upon the phospholipid molecules remaining in that particular shape that they are in. So if they come too far apart, let me use the other one, if they move too far apart, then the membrane will lose its partial permeability. It means that everything can run through it, and that is not going to be good for the cell if it moves too far apart at high temperatures. If it is too cold, low temperatures, and they come too close together, then it will become impermeable, and so tiny molecules like carbon dioxide, oxygen, and even water molecules that can move through this fluid, through the phospholipid membrane, they will not be able to do so if it comes too close together. So cholesterol is extremely important to maintain the balance of the membrane in the sense of making sure that it is not too fluid, meaning that everything can move through, and not too rigid that it prevents certain molecules from coming through. So please note that about cholesterol, it's important. Now the fluid mosaic model of the membrane just really refers to that the fact that the membrane is fluid, it's always in motion, right? um, and and that motion is basically based on the phospholipid layer by layer, right? They're not fixed like a nail something to a wall or anything like that. They have a slight movement going on, The mosaic basically describes that pattern of proteins distributed across the membrane, the phospholipid bilayer. So it looks like, if you see a mosaic, it has different little dots and little parts in it. So that's what the membrane, that's what the mosaic describes, the way the proteins are dispersed within the membrane. So fluid mosaic model is that description we use for the membrane because it actually resembles that. Alright, so let's now look at how stuff moves across the membrane, starting off with the most important process, diffusion. And just to understand diffusion in the beginning, particles are always in motions, and this is something that is important for us to understand. Particles always move. There's nothing that is basically stationary. Anything we consider stationary, it means that it is moving so slow that our eyes cannot detect it, and sometimes it may be moving so fast that we can't detect it either, but that's a discussion for another time. We're looking at particles always moving in all directions, spreading evenly into any space that they are found in. You have to prevent them from moving for them to remain stationary. Once there is more particles in one space, right, more than the other, then we say that there is a concentration gradient, which means if we have more particles in one area of a room and less in another area, then we're creating a concentration gradient. Now what diffusion is the net movement. When we say net, we mean that the majority of the particles, molecules or ions, they move down their concentration gradient until they're evenly spaced. So if we have more particles on one side of the room and less on the other, they're going to move from where there is more down to where there is less particles. So that's what we mean by moving down the concentration gradient. So many different particles are constantly moving and bouncing around on the membrane. So this is important. When we have the phospholipid membrane, particles bounce against the membrane. This is a random thing. Good. So that's why when we talk about diffusion going faster, we mean we increase the temperature, which means we increase the speed at which these particles move, right? However, if their particles have no charge and are very small, then they will be able to slip through the plasma membrane. So particles that are extremely small and have no charge can slip through the membrane. Note also particles like water, they are very small and they do have a charge, but they can move through the phospholipid bilayer as well. Alright, so examples of particles that are small and have no charge and can move through the membrane includes carbon dioxide and oxygen. Due to aerobic respiration, oxygen is constantly being used up, creating a lower concentration inside the cell compared to outside the cell. So there we get our concentration gradient. Alright, and if we get that concentration gradient, then it means that the oxygen will always diffuse down their concentration gradient, and hence they will diffuse into the cell. Good. This form of transport, we call it passive transport, right? So note also lipid soluble molecules, no matter how large, can pass through the membrane as well. And the reason for that is because they don't have a charge. Good, they're lipid soluble. So once they're soluble, then they're able to go through the membrane. Alright? Now facilitated diffusion, right, is where cells provide a special way for ions to pass through it. This is done using channel proteins or carrier proteins. Good. So these proteins span the membrane and are also called membrane protein. So if you look at this little diagram right here, what you can see is a channel protein is that little pore. So that protein that spans the entire membrane. Once the molecules continue to bounce around until they find that channel, then they will go through it. So basically it provides like a doorway. These molecules, sometimes you can have molecules that have that are large and are unable to go through the phospholipid bilayer, so they need a doorway or a pathway or a channel through which they can move across. So they will move around randomly on the membrane, constantly moving around until they bounce and find that hole or find that channel, right? And then they will go through. And this is the moving from where there is a lot of them to where there is less. So they're moving down their concentration gradient. In the second picture here, where we have carrier protein, the carrier protein is actually a carrier. So once a molecule fits into the passage or the crevice or the area where it is supposed to, then the carrier protein will actually either open and allow the molecule through, or it can flip over and bring the molecule in, whichever method it chooses, right? But usually it's not like the channel where it just moves through. It has to hold the molecule and bring it over into the cell. Alright, so the ions can therefore move through these channels down their concentration gradient, and this process is called facilitated diffusion. It is the same as simple diffusion, only exception is that the molecules needs to be facilitated across the membrane, and this is done using either a carrier protein or a channel protein. Facilitated diffusion, as I said before, just like simple diffusion, except right for the channel for them to bounce into. Now each channel is specific to a particular ion or molecule, right? The channel also has the ability to either open or close, prevent or allow certain substances into or out of the cell. Some channels can allow the movement of more than one substance in the same direction. We call that a SYMPORT channel. SYMPORT, SYMPORT, same direction, sim, good. So that means that the molecules move through in the same direction, while in opposite direction we call it an antiport. So you have some carrier proteins or channel proteins that we call them symport, means that it allows more than one substance to move through in the same direction, and you can have an antiport where you have opposite movement, right? Now comparing facilitated diffusion and simple diffusion, we're comparing based off 1, 2, 3, 4, 5 criteria in terms of the direction of movement. They both move down their diffusion gradient. Additionally, is there any requirement for additional energy? No. Both of them, they use the energy that is in their particles already, so they need no additional energy. So this is a passive transport. Both of them passive transport. Type of solute, any solute is possible with, and solute here means we're referring to the particles, ions, molecules that move randomly. It's any when it comes to simple diffusion. So any solute, any ion, any particle, any molecule can will be moved using simple diffusion. However, when it comes to facilitated diffusion, because the protein, the channel through which the molecule moves is specific to a particular ion, particle or molecule, then the diffusion, facilitated diffusion, will be specific to that molecule that the protein is specific to. Right, so channel proteins present are carrier proteins. They are not used in simple diffusion, but they are present for facilitated diffusion. The speed of diffusion here vary for both. Why we say vary for both? Because the speed of diffusion depends on the temperature under which it is occurring, and the temperature really determines the amount of energy each particle has. So if you add energy, then diffusion goes faster. If you remove energy, then it goes slower. So it really basically occurs the same. One point to note is that because facilitated diffusion means that you have to bounce and find a channel to go through, right? So while simple diffusion, you don't need no channel, you just move randomly from where there is a lot to where there's less. What in facilitated diffusion, yes, you're moving, but you need to find a channel. So to find a channel might slow down the process, as well as it can speed it up as well, depending if we're looking at a specific molecule. Alright, so that's why I put vary for both of them, because the conditions, once the conditions are right, then either can go fast. Alright, now look, let's move on to osmosis. Now water molecules are very small and therefore are able to pass through the bilayer by diffusing through small spaces between the phospholipid, right? They also move through channel proteins that are fixed open, and these are called aquaporins. So aquaporins are really some fixed open channels through which water molecules move. Alright, now we're going to make it a little bit more tricky for you now when we say that this movement of water down its concentration is and if you notice I have in brackets here water potential, because we're going to be using this term now. So I'm introducing this term to your water potential, right? So water moving down its concentration gradient is called osmosis. So when we're moving from where there's a lot of water molecules to where there's few water molecules, we call that osmosis. Is net movement of water down its concentration gradient. Now when we talk about concentration, we actually referring to solutes, and because there's no solutes when we're talking about water here, when we're talking about pure water molecules, we have to start use the term water potential now. So it is not accurate to describe the amount of water in present in a substance as being okay. So we are talking about water potential here. It is not accurate to describe the amount of water in something using the term concentration. This is because the term concentration is used to indicate the amount of solute present in a solution. So if you're talking about pure water, you can't say pure, or you should not say the pure water concentration of this or concentration of that, because concentration is referring to the amount of solute that is dissolved in a solvent, which usually is water. A more accurate term to describe the amount of water present in a solution is water potential, and this is the symbol here we use to represent water potential. Now, pure water at normal atmospheric pressure is equal to 0. Therefore, adding solutes to pure water will decrease the water potential to below 0, making it a negative value. See? So if you have a container of pure water, then once you add solutes, whether it be salt, sugar, sucrose, starch, anything you add to it will decrease the water potential and make it negative. So when you add water, you increase water potential, and then you go to the highest, which is pure water, equals 0. Hope you get that. So solutions containing a lot of water under pressure has a high water potential. A solution containing a lot of water dissolved, a lot of dissolved solutes and little water has low water potential. See that? So water under high pressure, any solution containing, so when it is a solution, you know, say it have a little bit of solute. A whole heap of water under pressure, it is high water potential. The highest water potential you can get to is 0, which means you have pure water. So a solution containing a lot of dissolved solutes, which means you have, if you're talking about a solution of salt and water, it means you have a whole heap of salt and a little bit of water, has low water potential. It's important that you understand that. Therefore, one can view water potential as the tendency now of water to move. So if you compare high water potential to low water potential, which one of them gonna have the higher tendency of water to move? It's gonna be the one with the high water potential. Hence, if you look at this diagram to your, should be your right, not sure, maybe your left, right? So here we have some solutes. This could be sugar, it could be salt, whichever. And over here we have pure water. Good. Now the net movement, according to your original understanding, the net flow of small water molecules would be towards the higher concentration of large molecules that's on the left here. And that's not the way I wanted to describe it. I wanted to describe it as where we have the higher water potential. The higher water potential would be on your right, and the lower water potential would be on your left. And the reason for that is because you have solutes on your left, so it means that the water potential is below 0. On your right, we don't have any solutes, which means that the water potential is closer to 0, if not 0. So the higher water potential would be on your right. So the water will move from a higher water potential to a lower water potential. And that is what we call osmosis, the net movement of water molecules from a high water potential to a low water potential. Now, if you recall your understanding as it relates to isotonic, hypertonic and hypotonic solutions, so a hypotonic solution is a solution where we have a very high water potential. So if you put an animal cell in a high water potential solution, it means that water going to move into that cell, because it's going to move from where it is high to low, because inside the animal cell you're going to have a low water potential compared to the environment which the cell is in. So a hypotonic solution, or another term we learn now, is a high water potential solution, right? If you put an animal cell in that solution where the water potential is high, it's going to immediately make the, when we compare the internal environment of the cell, the cytosol or the cytoplasm, the water potential is going to be low there compared to the environment. As a result, the water will move in and therefore expand. The cell will continue to expand and expand until eventually it will lyse or burst. Now, the opposite of that occurs when we put an animal cell in a very, very low water potential. So if you put it in a solution that has very, very low water potential, in a sense we're saying that the solution has more solute than solvent particles. Instantly, the cytosol or the cytoplasm of that cell is going to have a higher water potential than its environment. Therefore, water will move out. And the results of that is that the cell will become shriveled or shrinked, because it's like sucking air out of a balloon. Good. So the size of the cell will decrease, right? Cell membrane, all of those stuff will collapse because there's no the internal pressure of the cell is decreasing, just like when you take the air out of a balloon. In the opposite now, the internal pressure was increasing, right? And that now means that the cell expand until it bursts. In the middle here, we have an isotonic where the water potentials are both basically equal on both sides. So there's no net movement of water molecules out or in. Good. That's what we call an isotonic solution, where the water potential is similar to that of the environment in which the water potential in the cytoplasm of the cell is similar to that of the environment in which the cell is placed. So this is the effect that osmosis has on the animal cells. When it comes to the plant cells, it's a bit different, because we're going to eventually realize that there is a major difference between the plant and the animal cell, and that is the cell wall. Now we have two terms here in yellow, looking at them individually. First one, let's look at the plant cell that was placed in a hypotonic solution, because the cell is placed in distilled water. Distilled water is pure water. In other sense, we are saying that this cell is placed in a solution that has a very high water potential. As a result of that, water will move into the cell, because a cell cytoplasm will have a low water potential compared to its environment. As a result of that, the pressure in the cell increases, and the cytoplasm and the vacuole is going to expand, because it's like blowing air into the balloon. So it's expanding. Once it expands, it's going to reach what we call the cell wall. When it gets to the cell wall is called a wall because it acts like a wall. It's going to apply a force on the expanding cell membrane and the cell cytoplasm and the vacuole. It's going to do what Newton's third law says. For equal, it's going to give an equal reaction. So if the cytoplasm and the vacuole is being filled with water molecule and they are expanding, creating a pressure, causing the cell to swell, when it reaches the cell wall is going to exert an equal but opposite force on that cell membrane and the cell contents to keep the cell intact, so that the cell going to reach to, we call that some pressure is being applied. I'm going to give you the terms. When the cell reach into this state where it is stiffened but generally retains the shape, we call that cell turgid. Excuse me, turgid, right? The opposite of that occurs when we put the cell in a solution that has a very low water potential, which means that inside the cell you're going to have a higher water potential, and which also means that water is going to leave, because where the high water potential is the high tendency to move, and so it's going to move away to where there is less or a lower water potential. That moving away will cause the cell vacuole and the cell membrane, cytoplasm and so forth to shrink, like sucking air out of a balloon. So now the cell wall, the cell membrane and the vacuole going to decrease in size. That decrease is not going to just happen like flicking a switch on and off. It's going to be a process. So it's going to become flaccid, then plasmolyze. Plasmolyze is the extreme case, when actually the cell body shrinks and the cell membrane is pulled away or collapses away from the cell wall. So in the first case, where we have the turgid scenario, where the cell membrane and vacuole were being expanded and forced up onto the cell wall, The opposite occurs in plasmolysis, where the cell membrane actually collapses away from the cell wall. In this condition, we describe the cell as being plasmolysed, or we call the term plasmolysis, where the cell shrinks away from the cell wall. In between, we have the isotonic, where the solutions, the water potential on both sides, but inside the cell and outside the cell, are basically equal. Alright? But there's another term called flaccid or flaccidity. Flaccid describes the cell's condition moving towards the plasmolysis. Plasmolysis is the extreme case. Alright, so some books will label this cell in the middle as flaccid. It is not incorrect, but I want you to understand that when we say it's flaccid, it's because it is actually losing water. This is another extreme opposite now of flaccid. You gain too much water. Alright. So that should take care of, let me just touch back again. So if we don't understand water potential, we'd say water potential is a term we use to describe the tendency of water to move. High water potential means that you have a lot of water molecules in a space. And a low water potential means that you have more solute than water molecules in a space or a solution. Alright? And a water potential describes the tendency of the water to move. There is a lot more that can be said as it relates to water potential and pressure potential and so forth. What I can do is to add this term in here for turgidity. There's a term called turgor pressure. Turgor pressure would mean the pressure inside the cell. So that turgor pressure is the pressure being exerted by the water molecules when they come in to the cell and keep the cell swelling up. The turgor pressure is also the same pressure that the cell wall applies to the cell membrane, at the equal and opposite force in order to keep it intact. Right? So that's the internal pressure of the cell. In the plasmolysis case, turgor pressure is 0. And because turgor pressure is 0, the cell wall actually starts to fall away from the cell membrane actually starts to fall away from the cell wall. That's when turgor pressure is 0. Normal turgor pressure is here when you keep things normal. So you can look at this thing like inflating a tube in a tire. Right? So the cell membrane acts like the tube and the cell wall acts like the tire. Good. So when you have a flat tire, it's like plasmolysis. And when you have a very firm, stiff, hard tire, that's turgidity or turgid, right? So the internal pressure. There's another term called pressure potential as well. These terms can be used interchangeably. So we'll explain those as we move on. When we look at investigating the effects of different water potentials and plant cells, we'll do that investigation, so those terms will come out better there. Moving to active transport now. So far we have looked at 3 ways in which substances move down their concentration gradient from one side of the membrane to the other. And this method, we say it's passive because it doesn't require any extra energy. However, there are other ways and there are other substances that the cell needs to take in and to get rid of by moving them up or against their concentration gradient. So we used to hear moving down the concentration gradient, which is moving from where there is a lot to where there is less. But now we're talking about moving up the concentration gradient, which is moving them from where there is a little bit of them to where there is a whole heap of them. So we're actually going against the concentration gradient. To achieve this, the cell needs additional energy, and it's obvious we need energy to move things in against their normal tendency. Good, because we kind of reverse in what nature does. Naturally, we say that particles are always in random motion and they tend to spread themselves out evenly. And if there's an area where there's a lot of them to it, and another area where there is less, they will move randomly until they even out. But in this case now, active transport, we're talking moving these particles from where there's a few of them to where there's a lot of them. So we're going against the concentration gradient, or we're going up the concentration gradient. And we said to achieve this, we actually need some additional energy. So this is called active transport. Now, active transport is carried out by carrier proteins or transporter proteins in the membranes. Carrier proteins work in close association with ATP. You know that ATP is the cell currency for energy. ATP provides the energy for the carrier proteins to change their shape. So if you look at the passive transport here, we say that materials either move down through simple diffusion right through the membrane layer, or they go through a channel protein or a carrier protein. Good. All of these are passive transport. Over here we have active transport. Now we need some energy to get those ports open. Good. So an example now of a port that does active transport is, that sounds like we call that now, whatever it is, a port that deals with active transport. So sodium potassium pumps is one of the example. So here in step 1 we said that the cytoplasm, the sodium binds to the sodium potassium pump, and then after it binds, it stimulates the phosphorylation of by ATP. Phosphorylation just really here so means the release of energy. Alright, so now make the big word phosphorylation here so frighten you. When we move on to photosynthesis in unit 2 biology, you'll understand that phosphorylation really deals with the creation of energy or the release of energy. In this case, we're talking about the release of energy. 3 phosphorylation now causes the protein to change shape, change its conformation, expelling the sodium outside. Good. And the extracellular potassium now binds to the protein, triggering the release of a phosphate group, right? Loss of the phosphate group restores the protein's original shape, and therefore now it will release the potassium inside. So this is an example of a antiport. Is it? Yes. You have symport and antiport. So this potassium pump deals with 2 different substances moving in opposite directions. Remember we said that you have symport where the protein allow molecules to move in the same direction, and then you have antiport where you have them moving in opposite directions. So in this sodium potassium pump, we have 3 sodium molecules being expelled and 2 potassium molecules being taken inside of the cell. Sodium potassium pump, and it requires energy to do that. Right, so it's an example of antiport and symport. Good. So so this would be our example of active transport. There are other examples you could research and add as well. Yes. Now, exocytosis and endocytosis is basically the bulk movement of molecules. So cells can also move molecules or move bulk substances into or out of itself. Moving bulk substances out of the cell is called exit, exocytosis, and the substances to be released from the cell are contained in tiny membrane-bounded sacs called vesicles. So these vesicles actually contain the materials that are to be moved. So moving bulk substances into the cell is a way this way is called endocytosis. So this is basically the opposite of exocytosis. Right. Now, what you can do for review is to look at these questions. We can go through them right now. These can be sent to you in hard copy form. So figure 3 shows the structure. I hope you can see this clearly, as I know that the image might be a bit blurry. So I'll just read it. Figure 3.1 shows the structure of a fluid mosaic model of the plasma membrane. Use figure 3.1 to answer questions 1, 2, and 3. It actually says items. So you can see is an example here of the fluid mosaic model. And what you can do is try and identify some of the parts. They identify the phospholipids. They show you right here, this would be a channel protein, and this here would be a glycoprotein, which is also a peripheral protein. Why I call it a glycoprotein, you can see the carbohydrate chain spiking off the protein. Alright, so number 1 says the model of the plasma membrane is described as a fluid mosaic model. Why is it described as fluid? If we go through the answers here, it says due to the presence of water molecules within the membrane. Would that be the answer? Phospholipids can move around within their layer. That sounds it. Let's see if anything is close. Glycolipids can move around in the membrane. The membrane is able to change shape. So D is definitely out, because we're not talking about that, why it would be fluid. A is definitely out because we know that water just passes through the membrane. B and C are the 2 close answers, but let us examine B and C again. B says phospholipids can move around within their layer. The layer is actually the phospholipid forms the layer. So we can say that phospholipids move around in their layer. But if we say that glycolipids can move around in the membrane, which is the same as the carbohydrate attached to the membrane, these 2 are very close. So we can select the best or the most appropriate answer here would be the glycolipids can move around in the membrane, right, or within their layer. How can the layer of the membrane or plasma membrane that forms the outer layer of the cell be recognized? How can the layer of the plasma membrane that forms the outer layer of the cell be recognized? Once it comes to recognition here, we're going to be looking for presence of carbohydrate chains. Remember that these carbohydrate chains on the glycolipid and on the glycoproteins help in cell recognition. So once we see the presence of carbohydrate chains, then we are able to recognize the cell. Why are the phospholipids in the plasma membrane arranged in the membrane as shown in the figure above? Phospholipids have, let me read that again. Why are the phospholipids in the plasma membrane arranged in the membrane as shown in figure 3.1? Phospholipids have A, hydrophilic heads and point outwards to the water molecules. That is correct. Hydrophilic heads that point away from the water molecule. That's not correct. Hydrophobic tails that point away from water molecules. That is also hydrophobic heads. We don't have hydrophobic heads. We have hydrophobic tails, so that is incorrect. Hydrophobic tails that point inwards towards water molecules. That is incorrect. Hydrophobic tails don't point towards water but away. So the only correct answer here would be A, hydrophilic heads that point towards water molecules. Alright, question 4 says the presence of glycolipid molecules in the plasma membrane allows for. So the presence of glycolipid molecules present in the plasma membrane allows for, not for the active uptake of ions, Yes, could be for, yes, it can be for cell recognition. Let us check if there's any other answer there. Diffusion of glucose, no. The fluidity of membrane, no, that is cholesterol. So the correct answer here would be B for cell recognition. If you check back glycolipids function, you will find that there. Which of the following molecules within the plasma membrane act as a carrier molecule during active transport? It wouldn't be the carbohydrate, wouldn't be the lipid, and it would be the phosphate, but it would be the protein. So it can be a carrier protein. 6 says a sodium potassium pump is used to transport sodium and potassium across the plasma membrane. What role does ATP play in the transport of sodium and potassium ions? Let us see the answers here. 8 says ATP attaches to the transport molecule, causing a change in the shape. That sounds like the correct answer. Let's check if there is any other that could be correct as well. B says ATP attaches to the ions, energizing them. No. ATP phosphorylates the transport molecule. No, we don't do anything to the transport molecule. Causing a change in its shape. This sounds correct. Let me correct myself right here. The transport molecule is actually the transport protein or the carrier protein, right? So ATP phosphorylates the ion. So this answer is actually more correct than the first one that we chose up here, which says that it attaches to the transport molecule. But down here, a more precise and a more accurate answer is that the ATP phosphorylates the transport molecule, which means that it gives it energy, causing it to change its shape. So the correct answer here is C. Number 7, which process is responsible for the transport of large proteins out of cell? Large bulky proteins, large things, anything we're moving out of the cell is going to be exocytosis. Number 8, the ways in which materials are transported from cell to cell differs. Which transport mechanism is correctly matched to the features listed? So a tick for yes and an X for no. So we're looking at the transport mechanism. Requires concentration gradient, requires ATP, and requires a carrier molecule. So does diffusion require a concentration gradient? Yes. Does it require ATP? No. Does it require a carrier molecule? No. So A is out. Exocytosis requires a concentration gradient? No. Requires ATP? Yes. Requires a carrier molecule? No. Good, unless we consider the vesicle. But let's check back for B. Let's see if what other options we have here. C says for osmosis requires a concentration gradient. Actually, yes, but remember we said that we don't use concentration gradient, we use water potential. But the question is still relevant here. Requires ATP? No. Carrier molecule? No. Facilitated diffusion gradient? Yes. Energy? No. So this one is incorrect. A is incorrect. B and C are the two close answers. Let us examine them again. So exocytosis, we said that this requires energy and it doesn't require a carrier protein. But here, and it doesn't require a concentration gradient. Osmosis, it does require a concentration gradient, but I say that we use water potential when we're referring to it. So in a sense, this one is going to be incorrect and the correct answer is going to be B. C is going to be incorrect because here we require a difference in concentration. We call it water potential, the tendency of water to move. Alright, so that's why this one is incorrect, and then B is going to be the correct answer because we don't require any concentration gradient at all, and we don't require any carrier molecule. We only require energy. Number 9, for equal lengths of potato cylinders, four equal lengths of potato cylinders were weighed and placed in a sample of bottles Sample bottles of various concentration of sugar. Table shows the results of weighing the potato pieces before and 2 hours after being placed in the solutions. Which concentration of sucrose is nearest to the water potential of the potato cells? Let us look at this one. So solution A, before it is 50, afterwards it was 49, and this is in grams. So the cell weighed 50.2 grams before, and afterwards it weighed 49.3. That shows a decrease in mass, which is equivalent to a decrease in the amount of water in the cell. So water moved out of that cell, and so the mass got smaller. B says it was at 50.4 grams before, and then after 2 hours it went to 60.3. Now that shows that there was an increase in the mass, and therefore it meant that it was placed in a solution where it had a high water potential. So water moved into the cell and increased the mass. C, the cell had 50 grams, weighed 50 grams, and afterwards weighed 51. So that's an increase of 1 gram, which showed it is similar to A. Sorry, not A, B. There's an increase in mass, so it still meant that the water potential outside of the cell is higher, so the water moved into the cell. Let's look at D now. D at 50.3, and then now it's at 51. 51, which means that there's an increase of less than 1 gram. If there's an increase with less than 1 gram, then it means that this concentration is the closest. The water potential here is close because there's less water moving into the cell compared to the other two, where we had more water moving in either direction. Alright, let me check that again, because if we look back here at A, let's go back to A. A at 50.2, and then it went to 49.3. That is 0.9 difference. Am I correct in saying 0.9? Right? Because if you put 0.6 on this, 0.7 on this would give me 50, and then 2 more would make it 50.2. Right, so it's a 0.9 difference here. Down here we have 50.3, and then right, so there's a 0.7 difference down here. So the closer one would be D. The difference between these two is actually 0.7, and A it's 0.9. So these two are close, but the most accurate answer would be D. Alright. So final question, a strip of potato of known mass was placed in a 0.25 molar sucrose solution. The strip was removed from the solution and reweighed after 12 hours. Given the water potential of the solution to be negative 680 kilopascals. Don't worry about this. Kilopascals is just the units in which they measure the pressure. And the pressure here is the pressure potential. I tell you that these terms will become more clearer when we look at this investigation in the lab. What formula can be used to determine or to calculate the solute potential? I think I did not address this in the PowerPoint presentation, so it'd be unfair to actually give this question. But what it would mean is that the pressure potential plus the solute potential equals to the overall water potential. This question 10, I'll deal with this question when we meet in person. If not, I'll address it in my next presentation. So this is the end of the presentation. I hope that your understanding of these terms and concepts are improved. Please do not take all of what I said as the the end all or the be all. Please research and add, and if there's any errors that I need to correct, you can just let me know and I'll make the correction as long as it is valid. Alright guys, thank you very much. That's the end of my presentation right there.